My name is Felipe Polk, and I am an undergraduate working in Dr. Paulo Pita's Vascular Physiology Research Laboratory. The lab focuses on studying cerebral blood flow under normal and in disease states. The lab's main question is, how does Alzheimer's disease cause improper blood flow in the brain? Specifically, the lab conducts research to understand how specific receptors on endothelial cells could have their function diminished by Alzheimer's disease. It is hypothesized that cerebral amyloid angiopathy could interfere with the normal functioning of the NMDA receptor, whose proposed role is to propagate an upstream vasodilatory response to increase local blood flow in areas of the brain with increased neuronal activity. You're probably wondering, what is cerebral amyloid angiopathy and what is an NMDA receptor? A pathological hallmark of Alzheimer's disease is the production and accumulation of the amyloid beta protein in the brain. The buildup of beta amyloid around the walls of arterioles is known as cerebral amyloid angiopathy, and it is likely to play a role in the dysregulation of cerebral blood flow, which could lead to the loss of brain cells and cognitive decline. The NMDA receptor is a transmembrane ionotropic receptor that is highly permeable to calcium. They are found on endothelial cells and are activated by the neurotransmitter glutamate and D-serine. Upon activation, they allow calcium inside the endothelial cell, causing the release of vasodilatory factors such as nitric oxide. To answer the main question, the lab conducts experiments such as pressure myography and calcium imaging. In pressure myography, an intact segment of a pile artery is dissected from mice and is then cannulated at both ends, where it is tied off to create a closed circuit. The vessel is then pressurized at different physiological pressures or treated with different vasoactive compounds to determine in real time the function of the vessel, such as myogenic tone and myogenic reactivity. My current project in the lab is to determine if there are changes in NMDA receptor expression between pile arteries of wild-type and Alzheimer's mice. Data is collected through two-step real-time quantitative polymerase chain reaction. Fluorescence given by the polymerase chain reaction is measured to determine the amount of NMDA receptor expression in the pile artery samples of the wild-type and Alzheimer's mice. As of now, there have not been results generated from this ongoing experiment. Alzheimer's disease is the most common type of dementia. It is the sixth leading cause of death in the US. Today, it is estimated that about 5 million of Americans are living with Alzheimer's disease. One goal in Dr. Pita's research lab is to characterize how NMDA receptors could play a role in regulating cerebral blood flow, to study possible therapies that could improve their function. Working in the lab throughout the summer, I still have lingering questions. What specific interactions are there between amyloid beta and the NMDA receptor? Do lymphatic vessels in the brain also express NMDA receptors? How does cerebral amyloid angiopathy contribute to lymphatic dysfunction in the brain?